person who will be invited to begin with is Mr. Javad Sobhi. Academic experience and PhD researcher in the field of biotechnology. Attend the certificate for the training program on inquiry-based sciences education IBSE for science technology under the Auspices of UNESCO 2012. Lecture about Green Sciences of Iranian Civilization for annual celebration of the role model for South cooperation through science, technology and innovation, ISTIC, under Auspices of UNESCO 2013.
We want to talk about biotechnology, especially resist plant or resurrection plant. First of all, we wanted to take a trip to center of Iran, Yazd, around of Yazd. After this, we moved to west of Iran, Kermanshah, to find an approach to find how we can solve my problem. And after that, finally, I think this PowerPoint discuss about the material and method and how we can find new approach to the desertification. Go on. Desertification.
Far to the west of Iran lives a lineage of brave Persians. The people of Kermanshah have taken shots of momentous episodes in Iranian history.
Many historians regard Iran as the crossroad of civilizations. For thousands of years, numerous tribes, ethnic groups, and dynasties settled in this land, each one leaving behind unmatched elements of their culture and civilization, which they thought that would link them to eternity. Many of these elements have still remained in Iran until today. A perfect example is this World Heritage Site, the magnificent mountain of Bisotun in Kermanshah province, which carries nearly 3 millennia of history and civilization of Iran on its surface.
I walked up at this mountain for 5 years.
This gigantic
Exactly up at the historical places.
in the middle of the western part of Iran. The Bisotun is known as the epicenter of Paleolithic Iran, with caves that have been the habitat of first human residents of Iran as far as 70,000 years ago.
This plant here in Farvero, here. And this is not Photoshop, this is natural. All plant you see here, all of them resist, but not my plant. Not here. I had to climb mountain. We want to talk about this plant, especially about this plant. This is the, you saw them before, this resist plant. And you want to resist plant, main the plant can resist around drought. It has a tolerance about drought and else, but especially about drought tolerance in plant. The importance of research is about 110 countries have dry lands, and also 73% of dry land are affected by desertification. You know that around 50% of the world affected by desertification. This is so important. We can find solution for this problem, this major problem because you saw bad memories about animal, about plant, and about insect died in Yazd. We showed you. And you can see this, I think, Isfahan. Now come. Around of the world, similar us, they are having problem about water. And what's the resistance plant? My plant is able to revive and come back to life after 30 years. Can tolerance about water 30 years. This is so important. Which factor of this plant cause this effect? This is my plant. This is Fairmont and grow up of the mountain of my hometown, Kilimanjaro. This after drought stress, for example, for this situation, can tolerance for many years. For example, stand by for plant. And this plant, before stress, this right one, and after stress, drought stress, you can see this exactly resurrection plant, because again come back to life. And I wanted to talk about an introduction for you, because I think maybe it's the little information about biotechnology, and I want, and in brief, I want to talk about this. In biotechnology, we can produce a plant. For example, embryo or organ or callus or cell culture, we can produce a plant. The normal situation, only we can produce a plant by seed. But in biotechnology, we can produce many plants with many different sources. And about my field about resistant plant, first of all, we need explant. Explant means part of the plant, for example, root, stem, or leaves. After that, we need the plant cell culture media. This is need micronutrients or micronutrient and vitamins and growth regulator and media sterilization. For example, nitrogen, potassium, calcium, for macro, for mineral one, the micro one, zinc and at all. And for vitamin B1, B2 and else. For sterilization, we need autoclave and laminar flow. This we put my section here and for sterilization. And also we need this kind of microscope, centrifuge, and tissue culture room. Not difficult. And after that, we need isoelectric focusing, IEF, and electrophoresis, and this banding for understand before and after stress what happened exactly. And this gene gun to transfer gene to the other plant and others creation. But about discussion tolerance, we know the escape mechanism of drought. After that, extensive road system, for example, many strategies for plant. One of them, pasticcio, has an extensive root system. And for cactus, counter and reduce water loss from the plants. About my plants, evaluation of the discussion tolerant resurrection plant introduced. This is life cycle of my plant. You can see above the rock, after that under the leaf produce a spore, and here you can see heart shape, we call proton. After that embryo, young spurt at the end, finally you can see the adult one, formation. For understanding what happened exactly, what difference between the normal plant and the resist one, which difference can help us to tolerance about drought, which factor exactly? We must test many different topic to search and find exactly what happened about this plant. For this time, we compare the sensitive plant with resist one. And for a long time, we use drought stress. After that, record any evidence about changing. For example, changing in sugar, proline kind of protein, abscisic acid or ABA, or different medium and hormones such as growth regulator, auxin, cytokinin, gibberellin. And we must domestication of fern, because I had to go to mountain 3 times a week because to record my observation about this plant. This is so important to how we can planting this fern in my field, in my lab, for example. After that, we can thinking about how we can about water, relative water content, RWC, and finally about changing in genetic transformation of this plant. This is a microscope section as before and after stress. We cannot see any changing here. This is not answers my question because not meaning changing before and after stress. This xylem to like the pipe transfer water through the plant. But I think the important things about the stomach, the between 2 sections in the middle of the picture. About the stomach, so only under of the leaf this plant has a stomach. This opposite of the other plant, because one side, because to evaporation so useful this strategy. And this many kind of sample to the tissue culture of this plant. And this result, we did it about 3 kinds. For example, in absolute dark, we cannot see any growth. And the light, and also in the photo period, 3 conditions with 3 situations for about, I think, 6 situation about MS medium culture. You can see the result that is about dark we cannot see growth, about the concentration is important, and you can see this our plant grow out this situation. And this about use the growth regulator. We understood that growth regulator no need for this plant because, for example, as you can see this button of these pictures or resid or road very growth fast and not very good. And after that, we saw this on not a good shape, not hard shape. That's unusual picture for us. After that, about relative water's content. So you can see the sensitive one is, I think, blue color. The blue colors about water as a problem because need high level of water, but around you can see 8, for example, only 500 milligram or milliliter of water need for life. After that, we can see electrophoresis and protein bonding compare between the normal and the stress one. As you can see the many change. So we find 2 new protein appear after the stress. Meaning that 2 gene can introduce and the cause then the source of the resist plant against the drought. And this is so important. And this electrodes focusing, you can see again before stress and after stress. Other things about sensitive plant and tolerance plant. Compare protein between this we can find exactly what happened in the plant and which gene may be expression, and we can select this gene and transfer to other plant. For example, if you transfer a gene of this plant to other plant, we can grow any plant in the desert. This is so important because every year around 40 billion we waste produce of agriculture. Achievement of this research finding drought factor. Which factor exactly is reference to this resurrection plant and protein pattern of them, and find out is induction tolerance point in life cycle of this plant. And evidence about discussion tolerance, many articles about details. For example, you can see we publicate in Tarbiat Modares and other country at all. Thank you very much.